going through proteins and enzymes today. So we've got some exam style questions, not actual past paper questions, but they're exam style questions. And also we'll have a look at some of the activities in the active recall workbook as well. So let's jump straight into having a go at an exam question. I can see someone said they've got a test tomorrow. In fact, 2 people have tests tomorrow, so this will hopefully be great revision for you, depending on if this is the topic you've got your test on tomorrow. But if not, still all good revision. So let's have a look at this first question. Let's get the highlighter. So proteins are made up of the monomer amino acids, and you have to, so the command word is describe the amino acids in protein, or describe how the amino acids in a protein determine the structure of the final protein. Okay, so how do amino acids determine the final structure of a protein is basically our question. So first of all then, let's go to the pen. I'm going to add in that amino acids are bonded in a particular sequence. So that would be the first thing that I've got there. I'm also going to write that as the primary structure. Of a protein. That's going to be my first mark there. Then I'm going to go on to what happens next. So that primary structure is then going to have foldings to create the secondary structure. I'm going to say this goes on to form the secondary structure. Held in place by H bonds. And then the next thing is, I'm going to say there's further folding to make the unique 3D tertiary structure. So for this one, I'd also add in that is held in place by ionic and disulfide bonds as well, although it's only sometimes disulfide bonds. What I'm going to add in though is determine the final structure of the protein. So how it actually determines it is the sequence of amino acids determines where the bonds form. And therefore how it folds. So that's what I have for this one. I'm going to quickly jump onto the messages to see how people did in that. I think people are talking about all tests that they've got coming up. Any tips? Someone said, just watch Miss Estruch's videos. That helped me. So I'm guessing that must be someone that's a little bit further along. Okay, cool. No questions or any comments on that at the moment. So that is our first question to do with how the structure of proteins are going to be determined by the amino acid structure. I'm going to flip over now to this fill in the gaps to try and get you a bit more involved in the chat box, because if you're here and watching, I'm guessing it's because you are here to revise and improve your A-level biology. Best way to improve your long-term memory is to test yourself. So let's get it into the chat box, whether you're on TikTok, Instagram or YouTube. We've got fill in the gaps here linked to enzymes. This is A-level biology, but some of these, even if you're watching and doing GCSEs, then you may well be able to answer it based on that as well. So the part of the enzyme that attaches to a substrate to catalyze a reaction is known as the something something and it's unique in something due to the specific folding and bonding in the something structure of the protein. Let's go for this gap first of all. So the part of the enzyme that attaches to a substrate to catalyze a reaction is known as the something. Right, I can see Instagram got correct answers. TikTok, yes, amazing. I can see correct answers coming in on TikTok. How's YouTube coming along? YouTube, why have you been so quiet? I can't see any answers coming in, although I always forget this every week. YouTube has got about a 10-second delay. There we go. I can see the first answers come in. TikTok has no delay, and Instagram has maybe a 5-second delay, whereas YouTube's 10 seconds. Okay, so loads of you have correctly said active site. I can barely fit it in. There we go. And it's unique in something due to the specific folding and bonding in the something structure. Let's do both of those at the same time. So it's unique in something due to the something structure. What do we reckon? Oh, there we go. YouTube have got loads of answers. Sorry, YouTube. You just are getting the information a little bit later. That's why. I'm going to jump over to TikTok. Oh, yes, TikTok. Amazing. Loads of correct answers coming in. So we've got unique in shape. And this is the tertiary structure. So I hate how he does this. It always criticises my handwriting, like many of you do in the comments as well. Harsh, by the way, I see the comments. But yeah, you can barely see the writing. That should say tertiary structure. So due to this specific active site, enzymes can only attach to substrates that are something in shape. So something in shape, what do we reckon it should be? Someone said, where did this activity come from? This is actually my active recall workbook. So if you are interested, it's on my website, missestruch.co.uk. It's the active recall workbook. Or just send me a message on Instagram and I can send you the link. Complementary, yeah, but can you make sure you are definitely using the correct spelling of complementary? So compli with an E, complementary. If you spell it with an I there, that's when you say something nice to someone. So complementary with an E means like shapes fitting together, opposite shapes fitting together. So the enzyme and substrate then form a something complex. So this is a key marking point that is always worth a mark. Oh, someone said make your pen ink thinner. That is such an obvious solution. Thank you. Oh, I love it when I get the obvious answers coming in. Obviously, make my pen thinner, then it'll fit. Okay, enzyme, substrate, brilliant. Oh, look at that. Enzyme, substrate, complex. So much better. Thank you, TikTok. Right, let's have a look at the next bit. All reactions require a certain amount of something before they occur. And this is known as the something. What do we reckon for this? So they all need a certain amount of something. And this is known as the something. Yeah, well done. I can see correct answers coming in. It's the amount of energy before they occur. And that is known as, yeah, I can see loads of correct answers. Brilliant. It is the activation energy. Right, last one then. When enzymes attach to the substrate, they something the activation energy needed for the reaction to occur and therefore speed up the reaction. So what do we think for this one? Yeah, and you can say reduce or lower the activation energy. Brilliant. Okay, so I zoomed in a bit too much. I couldn't quite see it there on Instagram. Let's have a look at the next part. So we then need to describe the induced fit model, because at GCSE you need to know the oh, mine's gone blank. Lock and key model. You need to know the lock and key model, but at A level you actually have to know the induced fit model, and that is the now accepted model compared to lock and key. Now on different exam boards you still have to know the lock and key for some, others you don't. So for AQA, lock and key doesn't come up, but it does still come up a bit for OCR. But the widely accepted model is the induced fit. So this is usually a 2-mark question. You've got the image at the bottom to help you with this. So for the induced fit model, which I've said here, is usually a 2-mark question. What you could have then is, we can see that the substrate and active site are not completely complementary to start with. So that's the first mark. So are not completely complementary. But the substrate induces it to change shape and mold around it. This puts a strain on the bonds and lowers the activation energy. There we go. So the key thing that they'd be looking for is the fact that the active site and substrate are not completely complementary in shape, but when the substrate binds to the active site, that then causes it to change shape or mold around it, or induces it. And when it then molds around the substrate, it puts a strain on the bonds. It's kind of pulling it backwards, and that strain lowers the activation energy. So not are not completely complementary. Good spot. Right, so that's a little bit from the active recall workbook. Let's jump back to an exam style question. So we're now moving on to proteins, but from the point of view of protein synthesis. So this question is, mRNA is produced in the nucleus, and you have to describe the process of how it's produced. This is a 6-mark question. So we're describing the process of how mRNA is produced. So let's have a think about 6 marks for this, for how mRNA is produced. So to start with then, this is basically transcription. That's when we've got DNA being converted or copied into an mRNA copy. And one of the key things that they always look for in the mark scheme is naming the different enzymes involved and saying what the enzymes do. So that's where I'm going to start. Helicase, that's the first enzyme. Breaks hydrogen bonds between the double helix. So that now means we have 2 strands of DNA separated, but only 1 strand of DNA acts as a template. And that's a template for the copy of mRNA, the mRNA copy. So now we've got 1 strand as a template. We'll have RNA nucleotides in the nucleus. Align opposites, complementary bases. And the next thing then is the enzyme RNA polymerase, and that joins together the adjacent RNA nucleotides. So RNA polymerase joins together adjacent RNA nucleotides. I'm going to add in here that forms phosphodiester bonds. And it's a condensation reaction. Right, so let's see. We've got 1, 2, 3, 4, 5 bullet points. Last thing I'm going to add is, it said mRNA producing the nucleus. It's not said pre-mRNA, so I'm actually going to add in that the introns are removed from pre-mRNA to make RNA. So let's check. We have got the enzyme helicase. We've got the enzyme RNA polymerase. We've said what it does. It breaks the hydrogen bonds, and this one joins adjacent nucleotides together. So that could actually be 4 marks straight away. We've got that one strand acts as a template. We've got the RNA nucleotides align opposite complementary base pairs. Yes, we've definitely got 6 marks there. Let's go on to inhibitors then. So we're jumping around a bit here today, but it's to cover a range of different topics that I've seen requested. So now we've got competitive and non-competitive inhibitors. Just going to move you TikTok so you can see a little bit better. Oops, there we go. You can get this workbook. Yeah, so this is on my website, missestruch.co.uk. It's called the Active Recall Workbook. I think I need to just shift this slightly so Instagram can see. Because I'm filming this all on one camera now, it just means YouTube can always see. Whereas Instagram, I have to make sure that you can actually see as well. Do we need to know about ligase? For AQA, you don't learn about ligase except for in gene technologies, but you don't need to know about Okazaki fragments or ligase for AQA. So competitive inhibitors. Explain how a competitive inhibitor affects the rate of reaction. Let's go. Lowers the rate. Let's cross it out. Lowers the rate, but it's asked me to explain. That's a statement or description. So it lowers it because inhibitor is same/similar is what they always say on the mark scheme. So you can say either or. Inhibitor is same or similar shape to the substrate. Inhibitor can then bind to the active site. So you get fewer enzyme substrate complexes. Now, if you do add in enough of these substrates, this is why it's called a competitive inhibitor, it will compete with the inhibitor for the active site. So if you add in enough substrate, it outcompetes the inhibitor, can bind to the active site, and then the rate returns to normal. But that's where it's different from the non-competitive inhibitor. The non-competitive inhibitor, so I'm going to say inhibitor binds at the allosteric site. Now, you don't have to use that term allosteric site. What that means is it's a place on the enzyme that isn't the active site. So let's say this is your enzyme and that's the active site. An allosteric site would be a different location to the active site, so it could be there. So you can just describe that. You could say it binds to the enzyme, not at the active site, but at a different point. Or for fewer words, you can just say it binds to the allosteric site. This changes the shape of the enzyme because it's a protein. So when you push in that side, it distorts it. And therefore, the active site's shape. So then again, you get fewer enzyme substrate complexes. So that would be my descriptions. One thing I do just want to point out that AQA are really particular on the mark scheme to do with competitive inhibitors is these 2 bullet points are both worth a separate mark. So you would have to say the inhibitor is the same or similar shape to the substrate. And then for another mark, that means the inhibitor binds to the active site. So those are 2 different marking points. So making sure you definitely have one talking about the shape and one about it binding. Right, let's then have a look at this common graph that you get that you have to add your lines to. So draw the lines on the graph to show the rate of reaction with a competitive inhibitor compared to a non-competitive inhibitor. So I'm going to do my competitive inhibitor in red. The key things I'd be looking for, this would often be 2 marks. The competitive inhibitor should have a lower rate of reaction, but it ends at the same rate of reaction. Because when I say end, actually I should say at high concentrations of substrate, it will have the same rate of reaction, the same maximum rate. And that's because the substrate, if it's in a high enough concentration, it outcompetes the inhibitor. So you'd have a curve that looks something like that. So it's a lower rate of reaction at all substrate concentrations until you get to the highest of the substrate concentrations, and then it has the same end point. Next one then is we have to show the rate of the line for the rate of reaction against substrate concentration with a non-competitive inhibitor. And for this one, the rate of reaction will be lower at all substrate concentrations. So they'll be looking for a curve that is lower than the original blue, which shows with no inhibitor, but it then plateaus at a much lower rate of reaction, because even if you add more and more substrates, because it's not binding to the active site, it's not competing for that active site, you aren't going to get any increase in rate of reaction. Okay, let's go on to an exam question. So this time we're thinking about enzymes again. This is an application one. So we're told that metabolism in humans is controlled by many different enzymes. So metabolism in humans is controlled by many different enzymes. These chemical reactions occur at normal body temperature, and we need to explain why each different enzyme catalyzes a specific reaction and why enzymes enable chemical reactions to occur at normal body temperature. 5 marks. So this one is a 5-mark question. So let's see. We've got to, first we'll address this. So why is it catalyzes a particular reaction? So I'm going to start by saying black. Enzymes have a unique shaped active site, so combined with one substrate only, and therefore all catalyze specific reactions. I'm also going to add in that this is due to the tertiary structure. I could probably have split those bullet points up a bit more. So it can only bind with one substrate. I'm actually going to add in there with one substrate. I'm going to say due to complementary shapes. And that's about 3 marks now that addresses the first bit. Then, how does it enable them to occur at normal body temperature? This is basically explaining the induced fit model. So I'm going to say due to the induced fit model, because there'll be 1 mark for saying that, because they haven't mentioned anywhere the induced fit model. So due to the induced fit model, the activation energy is lowered. And that's actually probably going to be 2 marks. Induced fit model, lowered activation energy. I'm going to say due to the strain on bonds when the enzyme substrate complexes form. Okay, that is that exam question there. Let's go back to, I think I had one more page in this that I was going to have a go at with you, but have I done it already? Oh, it was here. We've kind of thought about these already, but this one is, I should have done this at the start, is to summarise the different structures, because this came up repeatedly in some of these exam questions. So for the primary structure, we can see here this diagram. The primary structure of a protein, that is the sequence, and you have to have the idea of sequence or order. Sequence of amino acids in a polypeptide. I'm going to add in held by peptide bonds. So each level that you have to describe, I'd be referring to bonds as well. Not for the quaternary, but for the primary, secondary, and tertiary I would. So sequence of amino acids, that's part 1. In a polypeptide, held by peptide bonds. Those are the 3 key concepts to include to make sure that you've got lots of key terms in your answer and you're being really specific, because we know how picky those exam mark schemes are. Then we have to describe the secondary structure. You've actually got a massive hint here, because this isn't an exam question. This is my active recall workbook, where it's bridging the gap between learning the information, testing you've understood it, and knowing key marking points, and then you go on to exam questions. So here, your first mark would be folding into an alpha helix, or it could be a beta pleated sheets. And then your second mark is to say what holds it in place, and that's what these pink dashed lines are showing. Hydrogen bonds held in place by hydrogen bonds. Right, the tertiary structure, this is now a 3 mark question. So this is now folded into a unique 3D shape. So it's kind of like the further folding, unique 3D shape. And then lastly, you actually have to name all the bonds involved. So the bonds that you could have are more hydrogen, ionic, and sometimes disulfide. If you are watching this with other exam boards, you have to know about the intermolecular forces as well, but you don't need to know that for AQA, you just need to know those 3. Lastly then, the quaternary structure. This is a 1 mark question. It's when you have more than 1 polypeptide chain. And the key example is just here, hemoglobin, because we've got 4 polypeptide chains. So there we go. That is our active recall workbook pages completed. Someone said, can you show it as a whole page, please? If I zoom out like that, it'll probably be quite small, but you might be able to screenshot it and then zoom in again in your own time. So there you go. Hopefully you can see that okay. Globular and fibrous proteins, a few of you were asking, that is not on the AQA spec. It is on the OCR spec. So you need to know about that for OCR. You don't need to know about this for AQA. Someone said, is a recording of this going to be uploaded? Yes, it gets uploaded to both my Instagram page and my YouTube channel. I probably won't be able to upload it for a couple of days. I normally do all the editing and upload on a Friday, but my children aren't at nursery tomorrow, so I won't be able to. So it'll probably go up on Monday. And also, as I said last week, I cut it up into lots of different TikToks, shorts, reels, so you can see a question at a time as well. Let's see, I think there was one more question. Yep, so here's our final question of the evening. Enzyme function can be reduced by 2 different types of inhibitors. Name each type. Oh, we should have done this one first because hopefully you remember this. We've just done a question on it. So you have to name each type of inhibitor and then describe how each reduces the activity. So this is where I was saying what I deliberately did for the active recall workbook design was I wanted it to bridge the gap between you learning the information and before you do the step up of exam questions. So that's why I had like these short answer questions, more guided, where it might be you've got the pictures to prompt you and things like that. So we've basically done this exam question for 5 marks was what we bullet pointed and thought about step by step here. So that's the point of the active recall workbook. It's to test yourself after lessons or to do as revision before you then move on to exam questions, because it bridges the gap. So hopefully when you then get to these long answer questions and other ones, it doesn't feel so overwhelming. So you just think, well, I just did that in that bullet point box and just transferring it here. So we have to name the 2 types of inhibitors. So let's do competitive up here. Competitive, and I'll do non-competitive down here. So that will be 2 marks straight away, or maybe it'd only be 1 mark, I don't know, but naming the 2 types. The rest of the marks will be for describing how each reduces enzyme activity. So for this one, I'm going to say inhibitor is the same or similar shape as substrate. So it binds to active site and that prevents enzyme substrate complexes forming. Non-competitive, that's when the inhibitor binds away from the active site or at the allosteric site. That causes a conformational, which basically means like a change in shape, conformational change in the protein. So active site changes shape. And then again, that means fewer enzyme substrate complexes. Mic drop, we're done. That is our our active recall workbook questions, our protein and enzyme exam questions, kind of bridging the gap between the 2. So hopefully you found it helpful. As I said, this does get uploaded onto my Instagram. So if you're watching on TikTok, then make sure you're following on Instagram as well, so you see the recording, or over on YouTube. And if you have any requests for topics, then let me know. I tend to just check it based on the comments that come up on the previous week's live. So if you do have any requests, then make sure you put it onto the recording of this live. Because last week I had loads of year 12s. I assume it was year 12, so they wanted more year 12 content. So that's why we had year 12s this week. But that is it for today. I hope you found it helpful, and I'll see you next Thursday, everyone.